Thank you so much. I've been on the job a mere 2 months now, and this is my first Celebrating Science benefit, and I'm absolutely delighted to be here and to welcome all of you this evening. Tonight's event has a special significance for Rockefeller since we're honoring our remarkable board chair, Russ Carson, and his equally remarkable family, Judy and Cecily and Edward and Madeline. We're recognizing them for their long and generous contributions and commitment to Rockefeller, as well as their numerous contributions to the arts, education, health, and medicine in New York City. I personally am indebted to Russ as well for chairing the search committee that brought my wife Deborah and me here to Rockefeller and to New York City, and we're just delighted to be here. I, of course, have long recognized the remarkable science that goes on at Rockefeller every day, but since arriving, I've been even more impressed by the remarkable and collegial community and the extraordinary sense of shared mission that extends throughout the Rockefeller community. I should note that while Edward and Madeline aren't here to celebrate with us tonight, they're having their own celebration following the recent birth of their first child, Walter Russell Carson, and we're so happy for them and for their new aunt and grandparents. Congratulations. The Carson family has been involved with Rockefeller for 25 years, and extraordinary philanthropists and ambassadors for our mission of science for the benefit of humanity. You'll hear more about them a little later this evening. Before introducing our featured speaker, my friend and Rockefeller University professor and physician scientist, Dr. Robert Darnell, I'd like to thank the entire benefit committee for making this evening such a remarkable success. In particular, I'd like to acknowledge our honorary chairs for their sponsorship of this event: Miriam and Sheldon Adelson, Catherine Farley and Jerry Spire, Marie Jose and Henry Kravis, Jim and Marilyn Simons, and Lulu and Tony Wong. Thank you so much for your incredible generosity. As also noted in the program, we've had an absolutely extraordinary benefit committee supporting tonight's event, and the list is far too long to read here, but we appreciate each and every one of you. The proceedings from tonight's event will provide really critical resources for the Rockefeller scientists who are transforming our understanding of human development and childhood diseases. Your gifts also help underwrite our really amazing science outreach program, which engages K through 12 students and teachers from around the city in the exciting world of hands-on scientific discovery. And I think this is a truly remarkable program. It really gives me now great pleasure to announce to you that as a consequence of your incredible generosity, that we have raised more than $2.6 million in tonight's benefit, an all-time record. So for those of you who have never been to Rockefeller University before, you may be wondering what we're all about. Well, Rockefeller is without question the world's leading biomedical research university. We're dedicated to conducting the most innovative science that improves the understanding of life for the benefit of humanity. We're organized around a very small number of faculty, currently 79 faculty. Each of these faculty report directly to the president. We don't have silos defined by departments. And this unusual structure enables us to be both nimble as well as cross-disciplinary. And I think one of the signatures of Rockefeller University is that each of our scientists sets truly audacious goals in their and do truly remarkable work every day. Our labs are engaged in neuroscience, immunology, biochemistry, structural biology, cell biology, and genetics. All of these bear on fundamental processes that underlie every human disease. And if you would like to learn more about research programs in disease or basic science, please come, feel free to speak to me or any of the other many Rockefeller faculty who are here in the audience tonight. Our unique approach to science has led to some of the most transformative contributions to biology and medicine. For example, just up the hill in the Rockefeller Hospital in 1944, Avery discovered that DNA is the genetic material, one of the most important discoveries in the history of biology. In fact, there have been 24 Nobel laureates affiliated with Rockefeller University in medicine and chemistry. It's somewhat sobering to contemplate that if Rockefeller were a country, we'd be tied with France for the number of Nobel Prizes, trailing only the rest of the United States, Germany, and the United Kingdom. We also have 22 winners of the Lasker Award, the leading scientific award in the United States. And I should note that this year, our own Charlie Rice was a recipient of the Lasker Award for his work that transformed the understanding of hepatitis C from a disease that killed 350,000 people without anyone even knowing what the cause was, to a disease that is now curable with very safe and non-toxic therapy in a short period of time. This has transformed this disease to one that can now be cured worldwide, and his recognition is a perfect example of basic science contributing to the transformation of millions of lives around the world. As many of you may know, tonight's benefit is sponsored by Rockefeller's Parents in Science initiative. This was created in 2007 so that parents and scientists would have a forum to discuss cutting-edge research pertaining to child and adolescent health and development. In addition to hosting lectures, the Parents in Science group, along with our science outreach team, sponsors our annual Science Saturday Festival on the first Saturday in May, so please mark your calendar for May 6, 2017. And now I'm delighted to introduce our speaker this evening, Bob Darnell, and his topic, Next Generation Genomics, Reinventing Medicine for You and Your Children. Bob is a world leader in the field of genetics and genomics. He's the Robert and Harriet Helbrun Professor and head of the Laboratory of Molecular Neuro-Oncology here at Rockefeller. He's also the founding director of the New York Genome Center. Bob has received many honors, including election to the National Academies of Sciences and the National Academy of Medicine. For those of you who may not know, the National Academies were founded during the Civil War by Abraham Lincoln in order to provide independent and authoritative scientific advice to the government and the nation. And the National Academies have done this over the last 150 years and is one of the highest honors that can be bestowed on a U.S. scientist. Bob is also a recipient of an NIH Director's Transformative Research Award. And on top of that, he's a wonderful colleague who is well appreciated and admired throughout the Rockefeller community. So after Bob's lecture, I'll join him on the stage and we'll have a brief conversation and we'll then open it to questions from the audience. But now please join me in welcoming Bob.
Thank you. Well, it is a daunting pleasure to speak to you tonight. This is both the most fun lecture I get to give periodically to an audience like this that's full of wise people of a great breadth of expertise. And it also makes it challenging in a good way. So it's daunting to take what we live in the weeds in as scientists and physicians and extrapolate from that what's important about what we're doing. So that's my goal here, is to make this both accessible and hopefully interesting. The topic deals specifically with kids and parents and in the context of genomics. So I just want to also mention that my work in this area has been robustly supported by the generous insight, wisdom, and wealth of the Carson family, and I'm extremely generous to get to know them and grateful for all of your support. So I put this picture up here in particular. So this is the Rockefeller University in the old days. This is, I think it used to be a farmland here, and York Ave didn't exist. But the first two buildings illustrate a key principle of Rockefeller, which is this is the science building, and opened in 1901. And this is the hospital building, opened in 1910, 9 years later. So the core of Rockefeller University was as a scientific clinical institution. And in fact, it was the first such institution in the world, led the way with this prescient insight that combining medicine and science could be of value as opposed to just pure academic science or pure physician-led observations. So in those days, the killer disease was pneumonia. 1 in 5 kids in this country died of pneumococcal pneumonia in the 1920s. This is a picture from a children's ward back then. So many of the scientists began to study pneumococcal pneumonia in particular. This was the work that Rick referred to by Avery, McCarty, and McLeod. And that's McCarty, sorry about that. And what they did was to take kids into the hospital who had pneumonia, get some specimens from them, some bacteria, streak them on plates, and they found some colonies of bacteria grew in very small colonies, some grew in these big, round, smooth colonies, and the discovery that was so critical was they found they could take a material out of this smooth, big colony, put it into the little colonies, and they would forever after grow big and smooth. That material turned out to be DNA. And so that was the discovery of DNA as the heritable material. And the real core of Rockefeller, in some ways then, is the onset of clinical genomics. So to push that forward, the university continued in its interest in clinical science and followed along the explosion of this area up into the 20th century. So that explosion really turned the page in science in the year 2000. So this is a pretty good headline for a scientist. The front page of the New York Times, "The Code of Human Life is Cracked," and Nicholas Wade wrote, "An achievement that represents a pinnacle of human self-knowledge." That's a good way to have your paper described. And really, it was Watson and Crick decided to crystallize this structure based on the observation that Avery, McCarty, and McLeod had made, that DNA was that transmissible agent, so they wanted to know what it was. Now, the first guy who got sequenced is this guy here, Craig Venter. And one of the promises at the time was that understanding the human genetic code would lead to transformations in what we can understand about disease and how to treat disease. And that promise got a little bit ahead of itself for a number of reasons. And those reasons are sort of on the second line here. The sequence took between 1990 and 2000 to complete. It's 10 years of work and cost $3 billion. So that's the kind of thing that could be done once, but only at great effort, great expense, and great time cost. So this isn't exactly an enterprise that could, on its own surface, transform science. And I'll tell you a little bit of the story of what's happened between the year 2000 and 2016. But one of the first things that happened is people stepped back and said, whoa, how are we going to try and apply this at scale to all the people in the world or the city or this auditorium? And what they did initially was to take this observation. It was $3 billion, was basically $1 per base pair. So to sequence this guy, he has 3 billion, like all of us do, 3 billion base pairs. So at $1 a base pair, it gets quite expensive. Now, the fact is nowadays at the New York Genome Center, which Russ and his family have supported, we can do that same sequencing for $3,000. So the cost has plummeted. But in the year 2000, they were still able to take advantage of the DNA sequencing to do a very crafty sort of geneticist experiment, where instead of taking other people and sequencing all 3 billion base pairs, they would sequence 1 out of every 10,000 base pairs as just little genetic markers, sort of like the petunia experiments of Mendel. A white flower, a red flower, the two together would make a pink flower. So they'd use individual nucleotides, the 1 in 10,000, as markers for what was happening in the genome. So that led to what's called the GWAS error, or DNA SNP chips, small nucleotide polymorphisms. And those 1 in 10,000 base pairs are scattered along the different chromosomes, such that each base pair could be a marker for the potential to develop a disease, like cardiovascular disease or brain disease and other diseases. So they weren't really ways of identifying what was wrong in a particular patient, but there were ways to identify the percentage chance that you might get one of these diseases if you had a particular conformation of these markers, a particular configuration. So that percentage risk is both powerful and has weaknesses to it. So I'll give you an example. This is the kind of approach that a company has used called 23andMe, and some of you may have had your sequences done with 23andMe. 23andMe, for several $100s, will sequence one nucleotide every 10,000 base pairs or so scattered across your genome and tell you what your potential risk for a disease is. So how do people understand what a potential risk is? Well, I think probably most of us intuitively get that because when you go to a doctor and get your cholesterol checked, you get, if you have a high cholesterol, the doctor tells you have an increased risk of cardiovascular disease. It doesn't mean you're going to have a heart attack that minute. It's a percentage play. And so 23andMe has used this. I had a colleague come to me earlier this year who's a 50-year-old man who developed myopathy, sore muscles, and a little bit of neuropathy, tingling in his hands and feet. Didn't know what caused it, and he's a very thoughtful man, is a very thoughtful man, I'm happy to say. The end of the story is good. So he went back and thought about what was wrong with him. He went to his doctor. His doctor had no idea. But he realized 6 months earlier he had been diagnosed with a high cholesterol and had started a drug, a statin, lovastatin, to lower his cholesterol. So he wondered whether that could relate to his muscle soreness. And he went and got a 23andMe test done and thought about it. Now, 23andMe doesn't actually tell you any answers. It gives you percentage risks if you want to go pour through your own data. And this guy, who's a computer scientist, did pour through his own data and realized that there are two genes, one on this chromosome and one on this chromosome, that increase your risk of getting a statin-induced myopathy, which is a rare phenomenon. It happens to about 1 in 1,000 people. And he looked at his 23andMe risk profile, and he had both of those gene variants. So his a priori risk would be high. But since he already had a myopathy, he said, maybe I'll stop this. So he did. He stopped his myopathy, got better. He told his doctor about it, and his doctor said, what, are you nuts? You've got high cholesterol. Start your statins again. So he did. And his muscle soreness came back. So he stopped it and quit it, and it went away for good. So it's a nice story. And it illustrates some of the power of this kind of approach of taking sparse data, something Jim Simons has taught me about, a mathematical approach to genomics, and getting something clinically useful extracted from it. But it also has limitations. If you had a point mutation in a single letter of the 3 billion base pairs, this approach would miss it completely. So let me fast forward then to about 2009. In 2009, the technology had gotten better, the sequencing was cheaper, and now we're able to, instead of looking at that very sparse data, look at each gene in the genome. So the way this works is kind of, I have the New York City apple analogy here. All 3 billion base pairs is the full apple of your genome. But the key parts of it, or at least an initial foray into the key parts of the genome, would be to look at 1% of the big apple, and that is the 1% that codes for the genes that themselves code for proteins. And the proteins are what makes up your muscles, your skin, your brain, your hair, and different proteins make the different cells in your body. So these are the different genes of your very smallest chromosome, which has 1,500 genes on it, sort of listed and crammed in to this page. And a very wonderful and famous scientist, who I can say without being too sycophantic, is sitting in this room, is Rick Lifton, who figured out that there's a way to get the sequence of each one of these genes and study them in great detail. It's a technology called exome capture. So what does it look like to study one of these? This is a disease gene that my lab has been interested in. And what Rick figured out is, how do you study these in detail? So this is the Fragile X gene. 3,000 base pairs of that gene code for one protein. And you can see it's a lot of letters there. GATC, it's not very interesting reading, although some scientists have claimed they've actually read it. The fact is, it illustrates sort of a needle in a haystack issue here, which is what we're trying to find in a patient who is sick compared to relatives, for example, who aren't sick, is literally one letter change in a big C like this. And that's a non-trivial problem, and I'm going to harp back on what this needle in the haystack issue is. One of the things here about this is what we would like to do is do this in a clinically relevant way. So the time becomes a big variable. If it takes you 3 years to figure this out and a patient is sick, it may be too late. So the technology has now improved to be able to sequence one gene like this within about 2 to 3 days. So one could take a sick child, sequence that child, and typically what we would do is also sequence the mom and the dad. And what we're looking for, the needle in the haystack, is a single nucleotide change. Here you can see the sequence is the same in the mom and the dad, but the child had a single nucleotide change, and this actually turns out to be in a real-life case. So this case was taken from Rick Lifton's work in 2009, where he first developed this technology to sequence all the genes, which number 23,000 in total, and look for changes in an affected child who had a syndrome that Rick was interested in, a dehydration-based syndrome, and compare that sequence to other members of the family. This is a family tree, and that's the proband, the affected child, a sibling that was affected, the parents, and here's a cousin who was affected. So doing this sort of technology, Rick was able to identify for the first time a single nucleotide that was changed in the affected people in the family that wasn't present in the unaffecteds. And that changed ultimately one amino acid and one protein that led to a diagnosis and a potential for treatment here. And Rick may talk about that more in the open session. Another example of this, which I also like, is an 18-month-old girl who had an onset of muscle weakness. This is a story provided for me by David Goldstein. A number of scientists have worked on this sort of syndrome here, where this can be a tragic but very difficult to diagnose problem. Lots of different things can cause such a phenomenon. And I have to say that in this case, this little girl I'm going to tell you about, the doctors thought they knew what it might be. They took their best guess, did a test, and it was wrong. And in that case, the patient, the child, the mom and the dad, and the doctors go on what I would call a diagnostic odyssey. And odyssey is maybe too strong a word, because Odysseus, when he went on his odyssey, at the end of the day had a good outcome. And a diagnostic odyssey may or may not have a good outcome. When you don't know what's wrong, the fact is 95% of the time, an answer never comes forth. However, and in this case, the child was being treated empirically for an autoimmune disease, which turns out she did not have. But now, fast forward, this is 2014, the sequencing was done of the same sort, using the same technology that Rick described. And in this case, another defect, a single nucleotide, was identified in SLC52A2. And you may have noticed Rick's case was SLC19-something, which just underscores how complicated this is. Solute transporter 52, not a particularly interesting gene, and one of 52 or more genes in a category. You never would have identified this a priori. But the gene sequencing told us what was wrong here. And this particular version of the solute transporter encodes for something, a protein that takes up a vitamin, vitamin B2, riboflavin. So this child effectively had a defect in the ability to absorb vitamin B2. And without identifying that, what inevitably happens in these children is they get weaker and weaker, and they get respiratory failure and die. So in this case, having identified the cause with genetics, it made sense to try treating the child with high-dose vitamin B2. And so these are the pictures courtesy from David at Columbia. And this is a picture of this little girl before her treatment with the vitamin. You can see her head is tilting to the side. She was so weak she could barely lift her head up. And she was confined to a wheelchair. So they gave her vitamin B2. 4 weeks later, here she is. You know, how can I resist showing this movie? It's just wonderful. And as a neurologist, I would still point out her arms are still weak, right? She's not moving her arms normally. However, she continued to get better, and 4 weeks later, she came back to the clinic. David says, high-fiving all of the doctors in the room. So this is a wonderful example of identification of disease, cause, understanding both from an academic and a clinical point of view, and driving towards a therapy. Now, all human disease genomics is not restricted to children. So Tom Maniotis, a friend and co-founder of the New York Genome Center with me, introduced me to Glenn Close. I'm not much of a moviegoer, and when the only movie I had known she did was 101 Dalmatians, so she wasn't pleased with me about my movie experience. But nonetheless, Glenn turns out to be the first woman who was ever sequenced. And in those days, things had gotten cheaper. It was only $100,000 to sequence her. And the reason she was motivated to do that was really altruistic. She had a sister and a nephew who were quite sick with neuropsychiatric disease. And they were not thought to be related in any way, but Glenn was prescient enough to say, well, maybe this is genetic, and maybe it would do some good if not only I got sequenced, but she paid for her sister to get sequenced and to her nephew. And lo and behold, to everyone's surprise, a genetic cause for their dysfunction was identified. And what the dysfunction was is they had severe neuropsychiatric disease. So her sister had mania, a severe form of manic depressive illness, very hyper, more so than me. And her nephew had schizophrenia. So when the sequencing was done, lo and behold, the defect was identified, but completely different from what I've shown you previously. So what they had in common, the sister and the nephew, was a duplication of a gene. So this isn't a single letter change. This an entire gene was duplicated. And it illustrates some interesting points. First of all, that too much of something isn't necessarily a good thing when you're a geneticist. And in fact, the basic principle here is that the genome is very finely tuned. So turning up too much or down too much expression of a particular gene, how much protein gets made, can cause big-time disease. And in this case, the extra copy of this gene encoded an enzyme called glycine decarboxylase, which means it's an enzyme that breaks down this amino acid glycine. And glycine is in all of our brains as a quieting molecule, sort of like a Valium. It's an inhibitory neurotransmitter. And this was cranking out too much of a thing that was breaking down and getting rid of the Valium equivalent, the glycine. So again, the same idea, glycine you can buy in a health food store. So they gave glycine to the sister and to the nephew. And Tom Insel, Glenn is very open about this story. And Tom Insel was the head of National Institute of Mental Health, says, and he describes this, it was like giving insulin to a diabetic. They gave glycine to these two individuals, and their neuropsychiatric disease just melted away. So they stopped the glycine, and they got worse. So they did it again, several times in a placebo-controlled trial. Or is this real? And it was real. It turns out to be that glycine basically cured the neuropsychiatric disease here. So this is extremely encouraging for a whole range of disease. And I'm showing these examples because they illustrate the good and the challenges in front of us for doing this at large scale for everybody. Everybody in this room, everybody in this country, everybody ultimately beyond the confines of the country. So part of the challenge in front of us is, I like this, it's complicated. And so just to run this through you, by you here, not through you, that would be too jargony, you would throw knives at me here. So as you know, DNA, these gene sequences get copied into RNA, and the RNA gets translated into the protein. The protein does the work in the cell. And what I've told you about so far is that we're able to, using Rick Lifton's technology, do this with the 1% of the genome that codes for the protein. So there's much more DNA present than codes for this protein. And in fact, if you look at the total DNA just to make one protein, it's a much longer piece than is needed. In fact, my dad was important in discovering that each gene, and he's sitting here in the front row or somewhere, there he is, good. I like to call him out because he made the effort to come here even though he's sick tonight, but not too sick, only a little sick. I know because I've examined him. So this is the copy of a gene. It's made much, much longer than the final form that gets translated into a protein. And what is beginning to be wondered about in the community now is whether the parts that code for the protein are part of the answer for understanding disease, But there are other parts, between these protein coding parts and between genes themselves, completely outside of the box here, that might be important in disease. And probably the story is that these are parts of the DNA that are like rheostats for turning on the expression level, too much or too little of a protein. And that harks back to the Glenn Close story. So there's a lot of interest in using the whole genome to get a more comprehensive understanding of disease, what's causing it, and how to treat it. So that creates a problem. So this is the 3,000 base pairs of one gene. If I was to try and illustrate for you what it would be to look at 3 billion base pairs, I would have to take this screen, which I carefully walked across earlier today and measured it out, and line this screen up to a second, third, fourth, and the screens would stretch from here to Las Vegas. So that's 3 billion base pairs. That's how long it would be. And again, in terms of the needle in the haystack problem, that becomes a more challenging and daunting task to really identify single nucleotide changes in such a long stretch of DNA. And to make it worse, as I mentioned, we typically would do this not only in an affected individual, but for example, in a mom and a dad. And to make it worse, that's one case. If I was to sequence everybody in this room, which we could do at the New York Genome Center now relatively cheaply and quickly, it would stretch this information from New York all the way out to Mars, okay? So that's a lot of stretches of GATC, probably enough to bore even the best scientist to try and figure out where the changes are here. So really what it means is we're in a different era right now. We're in the era of big machines, okay? Like it or not, the elephant in the room for genomics right now, or one of the elephants at least, a key bottleneck, is that we just can't do this in a way that's clinically meaningful, meaning for hundreds or even ultimately millions of patients without harnessing the power of computers. And I'm going to tell you just a little tale of that in the next 2 hours. No, you just flash me if I go. Kathy Van Kierie's watching, and Marty, if I go over time, I'll be shot at also. So but I just want to tell you a little some vignette here about machines and how they come online for thinking about genomics. So this is a hypothetical story, something also sort of important to realize. If you have 5 people who have breast cancer, they have 5 different diseases. Breast cancer is not a single disease. 5 children with autism, it's a rubric. We call it, doctors call it autism, but in fact, it's probably made up of many sub-diseases. The same with diabetes, the same with Lou Gehrig's disease, the same with almost any disease you can think of, except straightforward Mendelian genetics like hemophilia, and the kings and queens of England clearly passed that along, and their one gene is obviously the cause of disease, and that's something Rick has done 1,000 times. Rick has figured out 1,000 cases of Mendelian sort of genetics like that, things being passed in a simple way along the family. But many common diseases like diabetes, heart disease, cancer, and so on are very much more complicated. They don't run in a simple way in the family. At the same time, by sequencing them, we can begin using this sort of exome technology and ultimately whole genome technology to understand the root causes in occasional families. And what we found is that when you do this, the generic term, and I'm going to turn to autism as the best example here, begins to break down into different subcategories. So autism is a great example because Jim and Marilyn Simons have supported probably the best genetic exercise in the world right now for a single disease, and that is autism. So they have supported through the Simons Foundation the sequencing of 2,500 families, meaning that's 2,500 affected children with autism, which is a huge issue in society. As you know, it's about 1 in 86 children right now are diagnosed with autism. How many different diseases that consists of, we don't really know. But what the Simons Foundation supported was the sequencing of 2,500 kids, their healthy parents, and a healthy sibling. So 4 times for each family. And what happened in the course of that is that, the first family, a gene was identified, the 5th family, a gene was identified, the 2nd family, but the 3rd and the 4th had the same gene identified. So these genes started to pile up on top of each other, giving confidence, first of all, that they were on the right track, but also telling you that some genes might be a greater cause for causing autism than others. And something else important comes out of that, very important. And that is that if you do more of this clustering and the different kids and families fall into different groups, you start to see that those groups have meaning underneath them. So cluster 1 here, one of the first ones discovered, were genes involved in this rheostat that would turn genes up or down. That's called epigenetics. A second group were genes involved in how much protein is produced from an mRNA, how much translation goes on. And a third cluster had to do with excitability of the brain, sort of back to the Glenn Close story, how much inhibitory neurotransmission there was, how much excitatory transmission. That proper balance is critical. If that goes out of balance, that can also lead to autism. So this is both intellectually interesting. It takes us out of the era of wondering if mercury in a vaccine causes autism. No, it's a genetic disorder, and we can identify in many cases what the cause is. But also think about this from a pharmaceutical or treatment point of view. You now have the possibility, and David Alice spoke to you last year, those of you who were here, about epigenetics and the basic science underlying this that can drive the pharmaceutical industry to turn up or down these modifiers of gene expression. And the same with these other areas. So this is not only academically interesting, it's clinically extremely important. Now, at the same time, I would say this sort of paradigm that the Simons Foundation has put forth for autism, I think will extend well beyond autism for other complex, common diseases like cancer, ALS, and Alzheimer's disease, and almost anyone that we think of. So the complex, longer version of this is that using just the exomes, we seem to be hitting possibly a plateau. After having sequenced 2,500, we're kind of running out of gas in how many new genes we can identify. And what if the gene variants, the mutations, the things that are going wrong aren't in the exome? We'll never find it, or not easily. So we've turned now, again, with support both from the Simons Foundation, and I am the head of a National Institute of Health grant at the New York Genome Center, which is collaborative with Cold Spring Harbor and University of Washington and many, many other researchers interested in autism, to look at this whole genome and ask, are there variants that we can see in this more expensive and more complicated space? And the answer is beginning to look like, and this is collaboration with Mike Wigler from Cold Spring Harbor, myself and Ivan Yosifov, is looking at this this space outside of the 1%. We're starting to see hits that look like they may be informative, up to potentially an additional 25% of diagnoses already. And that means that more case clusters can be identified, more drivers for pharma to try and treat more kids. Now, the case cluster story itself is interesting to think about in this elephant in the room, machine language space, machine learning. So let's just take the theoretical case of, it could be autism, it could be any complex disease, where we see genes piling up and we'd like to cluster them into the proper bins they are, as defined by not a doctor and their clinical diagnosis at the bedside, but defined by the genes themselves. And I don't mean just the genes, really. What we're interested in doing is taking a group of patients who have a diagnosis like autism and ask, what are their genetics? What are the variants in other genes that may be playing into it? What environmental factors have they had? What habits do they have? What food do they eat? So for any disease, whether it's diabetes, Alzheimer's, autism, one would like to take complex data sets and see how these diseases bin into different clusters. So this is illustrated here in a program written by Rahul Satija, who's one of the junior faculty at the Genome Center, and NYU, he's a joint faculty there. This is a general concept here. So if you have a group of patients with a disease, name your favorite disease, you'd like to know how many different sub-diseases are there here. So he invented a machine learning tool called Seurat, and you'll see why. You get a sense of it here, that can take different patients with the same diagnosis at the bedside and say, do the genetics put them closer together than another group of patients with the same disease, and push them further apart. And so really the machine learns from all of that information how to segregate based on data, not intuition. And so this is the program running all of these different patients, and it takes them and says, which ones are closer to each other, which ones are farther apart, and pushes them apart and clusters them together. And this works so that, at least in theory, and I think in fact, this will be an extremely powerful way to try and cluster families together so the families can support each other, the pharmaceutical industry can look at the genes, and progress can be made in treatment. So I really love this, obviously. So I spent a lot of time making this slide, but I like the concept, and I love the name of the program, Surratt. So anyway, that's really concludes more or less what I wanted to say. I think we're at a spot now where we can think about clinically actionable genomics, not in the academic space, on 1 or 2 or 100 or 300 lucky individuals who get brought into a clinical trial, but in a way that can be brought to the entire community. Everybody in this room, everybody in New York City, with all of its genetic and ethnic diversity, which enriches our approach to genomics and is ethically the right way to do genomics. And some of the challenges and opportunities in doing this, I've broken down in my mind into a first mile, the middle distance, and the last mile. So the first mile means engaging patients into the genomic enterprise. They need to be educated about it, the good and the bad. What's the difference between 23andMe and whole exome sequencing? Well, whole exome sequencing can identify that riboflavin deficiency for the little girl. 23andMe cannot. It's not on that chip. So there are ways that this kind of tiered sequencing strategy needs to be thought through. This is cheap, this is medium, this is very expensive. So there's a cost-benefit analysis for each individual and for society to think through. It would be interesting to discuss. And there's issues about community engagement that are also ways that we can think about empowering the web. All the kids know how to do this, to communicate with each other and crowdsource. And that sort of idea can be brought to genomics as well. In the middle is the big chunk of the work to be done, which is still basic science, but I think to make it clinically actionable, there has to be a merging of basic science with clinically focused physician scientists, or at least scientists who think of this as the end, light at the end of the tunnel. And that, I think, will ultimately turn around the way we think about disease. So we won't think about disease as what the doctor defines it in the textbook when I was in medical school, but what the genetics tell us the sub-bins of disease are. And to really do this at scale, we're going to have to deal with the issue of becoming friends with machines. The collaboration has to be established, both to be able to do it at scale, at high quality, at speed, and it's something that's both a place like the Genome Center can help with, but also scientists will be an integral part of it, trying to teach machines and learn with them. And then the last mile is all of this is no good unless the information can be communicated back to the patient. It starts with the patient and ends with the patient. So the system has to be built in a way that there's an interchange the whole way, start to finish. So I'll just conclude by saying I love Rockefeller University. It is in my heart, and I've been here since 1992 as a physician scientist, because it's built at its core on the best-in-class science. Rockefeller, not to boast, is by 2 different metrics ranked as the #1 research institute in the world, on the planet Earth. And it is also, I guess that's redundant. Sometimes I wonder, next week I'll be done. And at the same time, there's no other place to go, so we're stuck. But at the same time, the basic science is merged at its core here with a clinical mission. That's our motto, and I'm delighted to be here. So thank you very much for your attention.
Thank you very much, Bob, for that really inspiring talk. We're now going to have a little impromptu conversation, and perhaps I'll lead off the discussion with a provocative question. Have you had your genome sequenced, and what do you consider to be the pros and cons of doing that? Maybe we can.
That is provocative, Rick. Yes, I have had my genome sequenced, and it was fascinating exercise. It showed me some of risk factors that I have. I have some obscure risk factor in a percentile basis for thyroid cancer, which nobody in my family has ever had. So I'm not sure what to make of that. I don't have risk factors for Alzheimer's, so I'm happy about that, but I may get demented anyway. So it's an interesting exercise right now, but the technology, in terms of being user-friendly, back and forth with an individual patient, is not in place enough to get as much out of it as is richly there in my DNA sequence.
So if you were giving advice to people in this room about how to deploy this technology, do you think we're at a point where everybody ought to be thinking about having their genome sequenced, or would you be thinking about, well, we'll sequence people if they have an illness that we suspect might have a genetic basis, or how do you think about this going forward, and how are we going to interpret the results?
I would say everybody in this room should get sequenced. That is my bottom line here, but I think there's two reasons to think in that vein. And of course, obviously, I'm being a little facetious because it's not really built yet. The system is not really built, but it needs to be built. And so, for example, there's two things that can happen. One is everybody in this room could benefit from learning what their risks are, their predispositions for things that might be coming in the future. It doesn't matter how old you are or how young. For example, there's an area called pharmacogenomics, where certain people, if they take common drugs like the statins, are at risk for getting a bad myopathy. So if you knew that ahead of time, you would avoid the myopathy. And in fact, some of these cases of myopathy are irreversible and severe. I know a colleague, one of our colleagues, has a severe irreversible myopathy. And that's true for about 400 to 500 different drugs right now. So why shouldn't we all get sequenced and prevent those problems? Some people have terrible reactions to anesthetics. If you knew about it ahead of time, we all may go under the knife. I did for my knee here, and I would like to know ahead of time what the risk is. So preventive medicine is something all of us could think about, and clearly that's true not only for us, but for our children. At the same time, if somebody in the family has a disease that is not diagnosed, it does stand a high chance of being looked at usefully by genetics. So I'm a big advocate for genetics, but I'm biased.
So I would generally agree with that. In my own view, I think if there's an undiagnosed disease, we're past the point where we wonder whether we might learn something by doing genome sequencing. And it's so inexpensive now compared to the cost of a day in an intensive care unit or an hour in an intensive care unit, that the returns on investment are quite high. In the general population, I've reviewed a large number of papers that said, we sequenced 50 ostensibly healthy 50-year-olds, and we're surprised to find that many of them had mutations that shouldn't have allowed them to live to age 50. And we then did extensive clinical evaluation of them, and were surprised to find that they didn't have those diseases at all. And of course, that's the challenge of having incomplete knowledge about how to interpret the sequence that we're seeing. So you're leading one of these large-scale projects for NIH to discover genes for complex traits. I wonder your thoughts about the power. We've modeled all of our initial studies on these diseases there where a single gene is necessary and sufficient to produce a trait. It may not be so simple when we get to more complicated disorders, even disorders that are more complicated than autism. I'm wondering how you're approaching this in your project.
Yeah, so I think you're making several great points here. First of all, it is premature to think about doing whole genome sequencing on everybody, because when I did it on myself, there were many, many more questions than there were answers to come out of that. On the other hand, I guess the issue, there's a very constrained space looking at 400 or 500 known markers of risk of drug reactions and so on, or carrier status for Tay-Sachs and things like that, where we do know a fair amount on a restricted space. So I think that's the kind of thing that could be brought out now for everybody. But I think on the other hand, you're right, understanding the complexity of diabetes or heart disease is a real research project right now. And I think it needs further technology in terms of understanding two approaches to it. One, which is an approach that is very viable and powerful, is to take a large cohort, a large group of people, 25,000 people who have the same diagnosis, and compare their genomes. Maybe it's their exomes, maybe some of them need their genome sequenced, and compare that to hundreds of thousands of people who don't have the disease. That is one strategy in genomics. And the other is this stratified approach, which is more like along the lines of what Mike Wigler has put forth for autism, but I think is also true. You know, you hear this story about BRCA1 and Mary Claire King and breast cancer. When they first sequenced breast cancer patients, it was a mess. I mean, nothing really came out of that because there's so many subtypes of breast cancer and so many causes of it, and it is difficult to see the signal from the noise there. What Mary Claire King did was to stratify breast cancer's cases into just where it was clearly running deeply in the family, and then take only those cases where it ran deeply in the family, presented very early, tragic cases in 20s and 30s. And that narrowed things down, confined the space so that the signal was easier to see. So I think that sort of stratified approach to genomics is an interesting adjunct to the big data approach, and they're not mutually exclusive. They should support each other.
To turn the conversation to another hot topic in biomedical research, in the last several years, we've seen cancer transformed in some cases from a uniformly fatal disease to one that is curable with immunotherapy. What do you think about the potential role for genetics and genomics helping to transform that from a therapy that helps a modest fraction of a modest number of cancers into an expanding universe?
Yeah. So I'm particularly interested in this. My lab has studied rare cases where people recognize, their immune system recognizes they have cancer and they get rid of it spontaneously. So there are interesting examples out there. Now some of these may be genetic. There are a group of small kids who have a disease called neuroblastoma, and they spontaneously get rid of it. Probably has a strong genetic component, which might fit into a traditional genetic strategy. In adults, it's thought to be more complicated and involve the immune system in ways that we're just beginning to understand. But I think one way to think about that is, first of all, more science needs to be done, that's for sure. And it needs to be in conjunction with clinicians. So when one takes a tumor out of a patient, regardless of what kind of tumor it is, what one sees when you look genetically at it is that it's many different kinds of cells in that tumor. And that includes different variations on the tumor itself and infiltrating cells, which can be white, and oftentimes are white blood cells that might be wanting to attack that tumor because it's got mutations in it, but are being prevented. So understanding the molecular biology, the molecules that are involved in that is just beginning right now, and that's the essence of immuno-oncology these days, is that some of those molecules have been identified, and identifying those does lead to a pharma target perspective, a way to try and attack cancer.
Terrific. I'd like to open it to the audience for questions. We have microphones, so question here in the third row from one of our friends.
Bob, when I first met you and you were going over to the Genome Center, you talked about getting information and then what would you do with it? And you used the example of meeting with a family where you had done a sequence and it showed something, not for certain. What would you tell them, especially if you could not do anything much about it or you didn't believe that you could? Where have you moved in the last 3.5 years on that subject, and how are you attacking it?
Yeah, great question. So really what you're asking about is, what do you do when you're the first person, for example, to have that mutation in SLC52A2? And the answer is we can't make a very definitive argument in that case because we don't necessarily know what SLC52A2 did for the first patient who got sequenced. So two things have to be done. One is the genomics has to be connected to the basic science, is a point that Tom Aniatis has impressed upon me, that the two don't go, they're not dissociated from each other. So somebody out there had actually been studying SLC52A2 and knew that it was a transporter of vitamin B2. So that information then gets put together to be useful. The other way that information gets put together to be useful is once more and more patients get sequenced and we all pool the data together as a community, all of a sudden you know, well, 12 people had this. Even if we didn't understand what SLC19 did, well, they all seem to respond to a particular therapy. So pooling data is an important aspect of genomics right now and helps answer that question, I think.
I'll just underscore Bob's point that if you want to understand your own genome, being able to compare it to everybody else's genome is the most critical element. And one of the things that we need to avoid is the idea that all of our genomes are so precious that they're going to be locked up in our own personal medical record and won't be able to share with the rest of the world in ways that are going to benefit all of us. So, yes, thank you.
Yeah, two-part question. First of all, what is the actual cost of having your genes sequenced? And secondly, what is the downside of having him? Are there any downsides in having him sequenced?
Good.
And thirdly, would identical twins have exactly the same sequence?
All three for good questions. The cost, what I was hinting at on that last slide is there's a tiering of sequencing that can be done. So 23andMe is about $200 right now. There are ways to make that cheaper, significantly cheaper, I think, and more powerful. So that's cheap, but the information is cheap, right, in the sense that you will miss things. And it doesn't give you definitive causes of disease, it gives you probabilities. So there's an important distinction there. On the other hand, it can be brought out to a wide, 23andMe has sequenced 1 million people so far. On the other hand, 23andMe also, it's not an academic enterprise where the data is pooled and accessible to everybody, and it's not connected to doctors and clinicians. So there isn't a back and forth for the individual patient. They're left on their own to interpret it. So for the money, you're kind of on your own there. Whole exome sequencing, I think, is a sweet spot right now because of Rick's technology, where it's relatively inexpensive and can be brought out in a clinically relevant way to a large group of society. And then whole genome is the gold standard for the moment of all 3 billion, and it's got technical reasons why it might be better in some ways than any of the other technologies, but it is more expensive. That's about, let's say, $3,000 per person compared to 1,000 compared to 200, something in that range. And the 1,000 can be brought down and the 200 can be brought down. Rick's got ways of making this less expensive. Your other parts of the questions. The downside is you might not want to know everything in your genome, right? You might not want to know everything in your genome. There could be little secrets in there, like some of them are too embarrassing to mention, like who is your father actually? You might want to know. You might not want to know that. Yeah, I have checked my dad, and he is my father. But there's about a 2% chance that he wasn't. So, and he never told me. So, but other things like diseases, for example, that you can't do anything about. Some people would want to know that, and some people not. So there needs to be a checkbox for each person, opt in or opt out for knowing everything or just the things that you could act upon.
One of the big challenges we have right now is this problem that we see variants that we have never seen before and don't know, even if it's in a gene that we know can be mutated to cause disease, we don't know the significance of that particular variant. And so the concern that raises is that we end up increasing the neurosis of the population rather than improving the health of the population. And this, of course, is not what we want to achieve.
And I just have to pick up on that, sorry, and then, which is we've undertaken a project, and others have, to sequence 100-year-olds, centenarians. And we found a couple of cases, and others, Carrie Stephenson and Decode Genetics have found others, where 100-year-olds are perfectly healthy, doing really pretty darn well, we should all be in that shape, right, have had exactly what Rick just described. Variants that are supposed to have caused Alzheimer's disease, and they were fine. So what it means, two things. One is we don't know as much as we think we do, and we have to have a little humility in the process here. And two, it's likely that what's really going on in individuals like that is that they not only had the risk for getting Alzheimer's, but they had a second gene or a third that were protecting them. So there's great science to be done here as well.
And the third part was whether identical twins would have exactly the same sequence.
Yes and no. How's that for a quick answer to a complicated question? Sorry, I'm not being facetious, but at a simple level, they had the exact same sequence. In the details, when cells divide, there's a 1 in 10 to the 9th rate of error that comes through. And after a lot of division, you can accumulate some errors.
Question right here.
I wondered, what is the procedure involved in sequencing? Is there a medical risk apart from the possible psychological risk?
Yeah, good question.
Disclosure.
Good question.
And then also, are there privacy issues? Can the patient opt for having the record locked in his or her own file, or is it inevitable that the findings would be generally known?
Yeah, so it's quite simple right now. We're doing a sequencing project here with Dana Orange, who might be in the audience somewhere, in patients with rheumatoid arthritis, and a single pinprick is enough blood to get your cell sequence, or spitting into a tube, which to me is harder because you have to spit and spit and spit for about 15 minutes, but you can do it that way. So it's not onerous to get your cell sequenced. The risks in terms of information are quite fascinating. You know, there's great care taken. There is some protection, the Genetic Non-Discrimination Act, which says that you can't discriminate on the basis of your genetics, but there are holes in that act that probably need to be plugged. It doesn't protect certain aspects of society in the elderly. So there are protections there, both technical in protecting the data from the Russians and
Bob, I think all of us should congratulate you for the successful launch of the Genome Center, along with Russ Carson, Jim Simons, Ivan Seidenberg, Tom Manialis.
I think they didn't hear the first part. You could say that again.
This man deserves congratulations.
Thank you, Harry.
But we've got to see that it gets ever more successful. What is your message to the average citizen interested, concerned, and excited about the Genome Center? What do you want them to do?
What I would like to do with it is to engage the community. So there's a component of education, just like we're doing here tonight, open forum, discussion, get people involved, even to the degree of sending in their samples for sequencing. But I don't think you can really engage them unless you set up a system where you can return what they want, which is not really your ancestry. That can be done, and that's cute and fun. But really, what people want to know is, what does their DNA tell them about their health and their future? And so I think there needs to be something that integrates the science, the machines, the person coming in, and what they want at the back end, which is clinical answers about what is their risk for them, or what solutions might be there for someone sick in their family.